In this section, we're going to look at the other type of chi-squared test that we learn in this class, which is called a goodness of fit test. And in a goodness of fit hypothesis test, we are determining whether the distribution of a particular data set fits or matches the distribution that we are expecting. And so, for example, to try and have us understand what this means, suppose a biologist is doing genetic testing and thinks that flower color will appear in a plant species with the following distribution: 25% red, 50% pink, and 25% white. The biologist can then collect data on the flowers actually observed to see if they follow this expected distribution. In order to perform a goodness of fit test, we need the test statistic. This is actually the same test statistic as we learned for our test for independence. We still are going to have observed data values, and we're still going to have expected data values. And so what we need to start with is what our null and our alternative hypotheses should look like, and then we will get into an example. So for a goodness of fit test, our null hypothesis is always going to be that the distribution is as expected. So null hypothesis is that the distribution is as expected. So again, no change or no difference from what we expect. The alternative, there's just going to be one alternative. So there's not the three choices. And the alternative is going to be just that the distribution differs from what was expected. Okay, or the distribution is not as expected. Okay, so those are going to be our null and our alternative hypotheses. Just like in our other test, our test for independence, we still have the rule that the expected value for each cell needs to be at least 5 in order to perform this test accurately. So we're going to go ahead and go into an example. And our example here, we're going to stick with that biologist example. So it says a biologist is studying genetics and thinks that flower color will appear in a plant species with the following distribution: 25% red, 50% pink, 25% white. And you may recognize that as outcome from like a Punnett square in biology. Flowers are grown in a controlled environment, and the biologist counts 36 red flowers, 88 pink flowers, and 30 white flowers. So those are the actual observed values. At the 5% significance level, does the data suggest the distribution of flower colors differs from what was expected? So the null hypothesis here is going to be that the distribution of flower colors is as expected. I'm going to be really specific here and say the distribution of flower colors is 25% red, 50% pink, and 25% white, only because I really want to make sure we understand that we are specifying that the whole distribution of the variable flower color is as we expect, so all 3 of those pieces. And the alternative then is that the distribution of flower colors is not as expected. So H sub A is that the distribution of flower colors is not as expected. That doesn't mean we are finding specifically where the difference is, just that it differs or it does not differ. Okay, so now that we have our null and our alternative hypotheses, I want to make sure also that we note our alpha level was 5%, so 0.05. Okay, As a reminder from the previous page, our test statistic, this is going to be a chi-squared test statistic, and is going to be the sum of the observed minus expected values squared over the expected. Okay. And so if we get started here, we have for red flowers, we observed 36. For pink flowers, we observed 88. And for white flowers, we observed 30. To find each expected value, as a reminder, we had a total of 36 plus 88 plus 30 flowers. So that's a total of 154 flowers. And we are expecting 25% of those to be red. So we need to find 25% of 154, which ends up being 38.5. We were expecting 50% to be pink, so 50% of 154, we would expect 77 pink. And then 25% again should have been white, and 25% of 154 is still 38.5. So we observed 36 red, we expected 38.5 red if 25% are red. Next thing we need is we need to find the observed minus expected value. So that's going to be the difference, 36 minus 38.5. That gives you negative 2.5. We then are going to square that value, which gives us a positive 6.25. And finally divide that by its expected value. So we're taking 6.25 and dividing it by 38.5, and you will get 0.1623. Okay, we're going to complete this process for each row. So observed minus expected, 88 minus 77 gives us 11. 11 squared is 121, and 121 divided by 77 is 1.571. And then last 30 minus 38.5 is a negative 8.5. Squared is 72.25. And divided by 38.5 is 1.877. And let's be clear here. If we add up all of those observed minus expected square over expected values, that gives us, so that's the sum of this last column here. That's 3.6103, which is our chi-squared test statistic. Okay. Our degrees of freedom, as a reminder, are the number of values of our variable minus 1. So we had 3 different possible values. So our degrees of freedom here are going to be 3 minus 1, which is 2. We can go ahead actually and look at how we would do this in the calculator and get that p-value. So in the calculator, we're going to stat, we're scrolling over to tests, and scrolling down to a chi-squared goodness of fit test, option D. You'll notice here observed should be list 1, that's our observed values, and expected is list 2. So we need to go enter those in. So first let's go to stat, edit. We're going to clear the first list and put in our observed values. So that's 36, 88, and 30. And then we're going to clear the second list and put in our expected values there. That's 38.5, 77, and 38.5. So now that we have observed values in list 1 and expected values in list 2, we'll go back to stat, tests, and remember it was option D. Observed are in list 1 now, expected are in list 2. Our degrees of freedom we need to change to 2, that's what we just found, and calculate. So you'll notice here for chi-squared, we get the same test statistic that we found by hand of 3.6103. I guess it's 0.039 if we were getting more precise here. And so our p-value is 0.1644. And we would then be comparing that p-value 0.1644 to our significance level alpha of 0.05. And since our p-value is greater than alpha, we do not reject the null hypothesis. And our conclusion then is at the 5% significance level, the data does not provide sufficient evidence to conclude the distribution of flower colors differs from what is expected. Okay, so we do not have sufficient evidence to conclude that the distribution of flower colors is different than the 25% red, 50% pink, and 25% white.